So, a couple housekeeping items. We do have a mastery quiz on Monday. I didn't bring clickers in today, so one finger means yes, two fingers mean no. Let's do a little poll. Question number 1. Can you use a page of notes for the mastery quiz? The answer is yes, you can. Okay? That will be standard operating procedure for these quizzes because the intent of the quiz is to try to get you to read the chapter. Will you necessarily understand everything that you read in the chapter? Maybe not. And if there's something that you don't understand, I always begin every class period by asking, do you have any questions? And what I'm hoping is that there will be things either from the previous class period that you didn't understand that you want to try to catch up on, or something from the reading. That's one of the reasons why I really encourage you to try to review your notes that you take in class prior to coming to the next class period. Because if you're taking notes in class, you might forget or not even remember that there was something that you were a little bit fuzzy about. But if you take some time and review those notes again before class, then you can go, yeah, I didn't understand that. Well, let's try to get those problems straightened out now rather than later. And particularly, I hope you don't wait until the night before the exam to find out that there was stuff you really didn't understand. Did you want to say anything, Larry, about the pre-lab quiz next week? Just so they can be aware of that?
The pre-lab quiz will be on the first lab, biological chemistry. Pay attention to the general goals of the lab, bold-faced words. You can use a page of notes, so if you outline the lab exercise, that will help you a lot. It'll probably be about 10 questions, which will either be fill in the blank or multiple choice. It shouldn't take more than 5 to 7 minutes, so be on time.
And the intention is really to have one of those before each of the labs, right?
Correct.
Okay. And these will not be in the Monday sessions, okay? The Monday sessions are for the reading from the textbook quizzes. So these are ones that you'll take when you walk into lab. And again, the purpose is to try to encourage you, or force you in some respects, to actually read the lab before you come in there. And so that's one of the reasons why we would let you have some notes that you take to just kind of refresh your memory.
So for our reading quizzes, so say like we're talking about chapter 3 on Monday and we have a reading quiz on Monday, would it be over chapter 3?
Yeah, I'll tell you specifically what the assignment is going to be for a reading quiz, at least by the Friday before. Sometimes it will be stuff we've already covered. Sometimes it'll be a chapter that we're getting ready to cover, but I'll make sure I let you know well ahead of time what the quiz is going to be on. Okay? So Monday, please read your lab before you come to class on Monday, because we're going to be talking about some of the things you're going to be doing in lab and kind of giving you a little kind of a preview of what's going to happen. The reason that we do that is because the labs, unlike your chemistry labs, are only 2-hour labs. And so we don't have a lot of time to give you instructions at the beginning of lab. So that's the purpose of these Monday sessions, is to kind of make sure, you know, this is what we're going to do. These are some of the kind of things to watch out for. These are some of the kind of stumbling blocks that people, you know, make mistakes with. And then again, the following week there might be some follow-up where we look at the data and say, okay, you know, here's some of the things that we found. Here's some of the interpretation of data that we did. So, but in addition to that, we'll probably have a little bit of lecture on Monday because that lab intro isn't going to take the full time. And the quiz, typically we leave you about 10 minutes at the end of the period for a reading quiz, or in this case, the mastery quiz. That way when you finish, you can go ahead and leave and just turn your quiz in. So any questions about Monday? Okay. So does Pluto have water?
Yeah.
Yes? You say that with certainty?
Yeah.
Why do you say that with certainty?
Somebody really big in space, I like that research.
Okay. Yeah, I was just reading online this morning that the New Horizons spacecraft, which is making its way out of the solar system, it just starts sending back pictures of the planet Pluto. And there is very strong evidence that there is ice on Pluto, water ice. It's kind of interesting because There's a lot of nitrogen and a lot of water on Pluto, but it's so cold that the nitrogen is frozen and is solid, and then these masses, kind of like glaciers of frozen water on Pluto, kind of float on top of the frozen ice, and so it's a very interesting planet. So if there's water there, think there's life on Pluto? I see some people saying maybe, some people saying no. People shaking your head no. How come? Planet? And if there's liquid water, who knows? Possibility of life. We're really finding that water is not quite as rare in our solar system as we once thought it was. We used to think pretty much Earth is the only planet that has liquid water, and we're finding out that that's not the case. That's particularly important for Mars. Anybody know why? Why is it important that there's some water on Mars?
Isn't it like beneath the soil? They've kind of been finding it like coming up and then freezing. Like
Yeah. One of the reasons why scientists are excited about that is because we're planning eventually a trip to Mars. Anybody seen The Martian? Yeah, sort of. Eventually planning to visit Mars. And the presence of water gives us a potential source of fuel. Okay? Because you can split water into hydrogen and oxygen, and the oxygen can obviously be breathed, and the hydrogen can be used as fuel. So the presence of water is pretty important. Okay, so what I want to talk about today a little bit is this really important concept of pH. Because I briefly mentioned the other day that pH is going to be really relevant when you start talking about biological molecules, especially proteins and nucleic acids, and their structure. So, what do you know about pH? What is pH?
Well, you want to keep it at a fairly normal level because it's very sensitive and the changes that can basically affect the structure and function.
Okay, it can affect the structure and function of molecules, and we'll talk about that. And you introduced this concept of levels. Expand on that a little bit more. What do we mean when we start talking about pH levels?
Lower levels are acidic and higher levels are basic.
Okay, so we have low levels of pH that are acidic, and high levels of pH, which are basic. And the statement was made is that we tend to try to keep things biologically kind of in this middle, in that state where it's neither too acidic or too basic. There's a scale associated with pH. What's the scale? What's the range of the scale? What's the most acidic? Most acidic. Most acidic is pH 0. What's the most basic? 14. So neutrality falls pretty much right in the middle. Okay? One of the things that we're going to see about the pH scale is that it is a logarithmic scale. So that's going to have some implications in terms of how many hydrogen ions there are in a given solution and how that is calculated. How did we define acids? What did we say acids were? They're molecules that when they dissociate release hydrogen ions, or what's another name for hydrogen ions? Protons. Okay? So at pH 0, there's going to be a lot of protons present, right? What are bases?
They dissociate.
They're molecules that when they dissociate produce the hydroxide ions. And so at high pHs, there's going to be a high concentration of hydroxide ions. Are there going to be many protons present at pH 14? No. So what's the relationship of hydroxide ions and protons at pH 7? They're equal, okay? So here, the concentration of hydrogen ions is equal to the concentration of the hydroxide ions. Does anybody know what pH actually is? what the definition of pH is? And yes, it's a measure of acidity or basicity, but what actually is pH? pH is the minus log of the hydrogen ion concentration of a solution. We said that water dissociated at not a very fast rate. At any particular given time, if you have a big container of water, only 1 out of about every 554 million molecules of water would actually have dissociated. And you remember what dissociation refers to? Here, that water molecule would have spontaneously split into its hydroxide ions and its hydrogen ions. Okay? So equilibrium would tend to be far in this particular direction, and very few of these molecules would have dissociated, but there would be some small finite amount of hydroxide ions and protons present in a solution of just pure natural water. At equilibrium, the hydrogen ion concentration is equal to 1 times 10 to the minus 7th molar. So what's the hydroxide ion concentration going to be? The same. So why do we assign a value of pH 7 to pure water? Because pH is the minus log of the hydrogen ion concentration, and pure water produces this tiny, tiny amount of hydrogen ions by its natural dissociation. The minus log of 1 times 10 to the minus 7 is 7. Okay? So the definition of pH tells us how many hydrogen ions there are in a solution in a given situation. If we start adding acids to water, then those acids, being proton donors, they release protons when they dissociate in water, will increase the hydrogen ion concentration. And therefore, this number will get greater. You might have 1 times 10 to the minus 5th. That's a bigger number than 1 times 10 to the minus 7th, right? And the pH of that solution of 1 times 10 to the minus 5th would be 5. It would be more acidic. When we get down to pH 0, the hydrogen ion concentration is 1 molar. When we get to pH 14, the hydrogen ion concentration is 1 times 10 to the minus 14th molar. Very, very tiny amounts. That makes sense? Okay, and because the pH scale is logarithmic, if we compare 3 solutions, pH 5, 6, and 7, which one has the most hydrogen ions? pH 5, 6, or 7? pH 5 does. It's most acidic. Its hydrogen ion concentration is 1 times 10 to the minus 5th. This one's 1 times 10 to the minus 6th. It's smaller. Another 0 added on. This one's 1 times 10 to the minus 7th. So what's the difference in hydrogen ion concentration between pH 6 and 7? It's a 10-fold difference, right? What's the hydrogen ion concentration difference between pH 5 and pH 7? It's 100 times different. What's the pH difference between pH 4 and 7? 1,000 times different. Okay, so it's that logarithmic scale that is the basis of pH. Well, okay, that might just seem kind of all esoteric, and what's that really have to do with anything? This just shows you a pH scale superimposed on some various fluids that you might be familiar with. Battery acid has a very low pH, very high hydrogen ion concentration. Household bleach very high pH, very low hydrogen ion concentration. One of the things we're going to consider here are some biological fluids like human blood. Human blood has a pH of about 7.4. And one of the things that you'll find out if you get involved in medicine at all is that if the pH of human blood starts becoming too acidic or too basic, that's a problem. And it can be lethal. One of the more common situations that can happen medically is that the blood can become too acidic. You have acidosis. And if that happens, that is really a life-threatening situation. Why? Why is acidosis so critical? Why do we need to correct that anomaly if we can? Well, living organisms typically exist in an aqueous environment or a water environment. You might say to yourself, well, wait a minute, we're not walking around in water right now, we're walking around in air. But we're multicellular. And the individual cells that make up our body are bathed by fluids surrounding the cells. There's tissue fluids that surround the cells. Blood cells are floating around in an aqueous environment. Blood's about 90% water. The pH of that fluid influences the charge of molecules that are present in the fluid. So pH influences charge. Anybody know what kind of a molecule that is? Good precursor for your quiz on Monday.
Amino acid.
It's an amino acid, okay? You've got this central carbon. You've got this functional group right here. What's that functional group called? You know? It's a carboxyl group. That is where the acid portion of the name amino acid is derived from. This is an acidic group. This functional group, you may know? Amine or amino group, okay? Well, this particular molecule, as I've shown it right here, is occurring in kind of a neutral state. There's no net charge on it. But it's also possible that that carboxyl group can become ionized and have a negative charge. It's also possible that this amino group can gain another proton and have a plus charge. These R groups, remember each of the different amino acids is characterized by a unique R group, kind of the wild card. And those R groups can become charged as well. Sometimes they carry a positive charge, sometimes they carry a negative charge, sometimes they have no charge. Well, what determines whether these particular either R groups or functional groups have a charge or don't have a charge? It's really dependent upon the pH of the environment. At low pH, this proton tends to stick onto the carboxyl group. At high pH, this group tends to become ionized. So if you start altering the pH of the environment, adding acid or base to the solution, you can change the charge on the amino acid. Why might that be significant to a protein if you start changing the charge on the amino acids? Yeah.
So basic structure function, change the structure.
Yeah. Basically, you're going to change the structure. Do you remember that there are several levels of organization of protein structure? Proteins are kind of fun. They've got all these different levels of organization. The most basic level of organization is called what? Do you remember? Primary structure. And what does primary structure consist of? It is the kind and order of amino acids that are present in that polypeptide chain. So it basically defines what the sequence of amino acids are. Okay? So if this chord right here represents a polypeptide chain, a bunch of amino acids strung together, is this typically what the protein looks like in vivo in the cell? Is it just this long linear chain? No, what's it do? It tends to fold up, right? And one of the levels of organization, called secondary structure, has to do with hydrogen bonding. And that has to do with forming structures called helix or beta-pleated sheet. Another level of organization called tertiary structure really has a lot to do with how those R groups interact with one another. And a positive charge on one R group might interact with a negative charge on another R group in the polypeptide chain, and therefore there would be an attraction between those two. And that would help to define how it folded. Well, what would happen is if we had at a particular pH, let's say pH 7, this R group was positively charged and this R group was negatively charged. What if we lowered the pH to pH 6? This one still might be positively charged, but now this one becomes positively charged. Are they going to attract each other now? No, they're going to repel. And what's that going to do? That's going to change the way that that polypeptide chain folds up. So we can, by changing the pH of a solution, change the tertiary structure of a polypeptide chain. And if we change the tertiary structure, what else do we change? The function. Well, how many times I will say that this semester? Structure determines function. And that's particularly true in the case of biological molecules. So even minor alterations in pH can cause proteins to unfold and lose their function. So, why is that relevant to blood? Why do we need to try to keep blood at a pH of about 7.4? Are there any important proteins in blood? Yeah, there's actually a lot of important proteins in blood. What's one really important one that you're probably aware of? Hemoglobin. Okay? Is the hemoglobin floating around just in the fluid of blood? Where do you find hemoglobin usually? It's inside of one of the types of blood cells, right? The red blood cells, the erythrocytes. As a matter of fact, red blood cells are basically just membrane-bound sacs of hemoglobin. What's that hemoglobin doing? What's its purpose? Carrying oxygen. Okay? When the hemoglobin is transported by the red blood cells to the lungs, it becomes oxygenated. When it then becomes transported to the tissues, it becomes deoxygenated, and it dumps off that oxygen. And then it goes back to the lungs again and becomes oxygenated again. That particular function is dependent upon a particular shape of hemoglobin. And actually, hemoglobin is kind of an interesting protein. You may know how many polypeptide chains hemoglobin is made of. It's not actually a single polypeptide chain. It's actually 4 polypeptide chains. There's 2 different types. They're called alpha and beta chains. So there are 2 alpha chains and 2 beta chains. Do you remember what a protein is called when it has a structure that is determined by several interacting polypeptide chains? That's that highest level of organization. Quaternary structure. Yeah, hemoglobin has quaternary structure. Well, the bottom line is that if you change the pH of blood, it slightly changes the structure of those alpha and beta chains. And they have a different shape. And at that altered shape, hemoglobin does not behave properly in terms of being able to pick up oxygen when it's supposed to pick it up and to dump off oxygen when it's supposed to. And so if the blood becomes too acidic, it just messes up that whole dynamic and you can't deliver oxygen to your cells. It's not released by the hemoglobin into the cells where it's needed. And why do the cells need oxygen? The biochemical process is using the oxygen. Aerobic cellular respiration, which is something that we'll talk about later on. And what does aerobic cellular respiration produce? Your cells absolutely need ATP, because cells need ATP to do work. And cells cannot stay alive without doing work. We'll talk about that later on in the semester when we talk about some thermodynamics. Fortunately for us, the body has some protective mechanisms associated with it that help to ensure that the blood maintains that desired level of about pH 7.4. And one of the things that helps to ensure this is a built-in system in the blood called a buffering system. What do buffers do?
They basically have an effect on the concentration of hydrogen ions
And hydroxide too, yeah. Yep. Buffering solutions tend to maintain a constant pH, even if you're adding additional acids or bases to the solution. Okay? So they're kind of mechanisms that assure that, within limits at least, the pH of a solution maintains fairly constant, even if you pour in a little bit of acid, or even if you pour in a little bit of base. Okay? One of the buffering systems that's really important in the blood is a buffering system called the bicarbonate buffering system. A buffer system consists of two things. It consists of a weak acid and the salt of a weak acid. Anybody know what H2CO3 is? What kind of acid? It's an acid called carbonic acid. You may know what NaHCO3 is? Sodium carbonate. Okay? So the presence of this carbonic acid and sodium carbonate in the blood helps to ensure that it remains at a constant pH level of about 7.4. How does it do that? Well, if you add small amounts of acid to that bicarbonate buffering solution, the base interacts with the acid and tends to neutralize it. If you add small amounts of base, the bicarbonate portion of the buffering system basically kind of soaks up that base and neutralizes it, maintaining a relatively constant level. Now, buffer systems are not indestructible. They can be overwhelmed. And if you start adding too much acid to a buffering system, you can overwhelm the ability of that system to resist the change of pH. And that's what happens sometimes in acidotic conditions in the blood. As the bicarbonate buffering system gets overwhelmed, too much acid is being added to the blood for a variety of reasons we don't have time to talk about right now. That's the province of physiology. But then the pH of the blood starts to drop. And then the charge on those hemoglobin R groups starts changing. Some of them go from positive to negative or negative to positive. And that interferes with the interaction of the R groups, which interferes with the tertiary structure of those polypeptide chains, which ultimately affects the function of that hemoglobin molecule. So this whole idea of structure determined function and pH having an effect on the structure of molecules is something that's really, really important in biological systems. So we'll come back there later on. Okay, so that's pretty much what you need to know as far as Chapter 2 is concerned. So let's move on and start introducing Chapter 3. Now you might say to yourself, well, chapter 3 is kind of the province of the mastery quiz. And I've already said, this is stuff you should already kind of know. And that is true. In chapter 3, there's really 3 important kind of subtopics within that chapter. And one of them is the importance of carbon. And we will talk about that a little bit in just a second. The second thing that we're going to talk about is functional groups. And then the third part of the chapter is the part of the chapter you're going to be responsible for on your mastery quiz. And that is these principles of polymers, the biological molecules. Okay? The carbohydrates, the proteins, the lipids, the nucleic acids. So prior to the quiz, we're going to kind of run through some of this stuff. And this will be material you'll be responsible for on your first lecture exam. This information is the biological molecules, and that's the province of the mastery quiz. And once you pass the mastery quiz, this won't be on your lecture material. Okay? Does that make sense? So the lecture material for your first exam is going to start with water, and then it will include the stuff we're going to begin right now, carbon and functional groups. But this won't be on it. Then the next topic is going to be cell structure. Okay? Okay, well, you probably all heard the statement before that life is carbon-based. As it turns out, there are some elements that are particularly crucial in terms of living organisms. They include carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus. I like to call those the big 6. And basically, these are the elemental materials out of which all of the molecules that life is based upon are constructed. Now, we're going to assume that you know really basic information about bonding and how carbon can bond to hydrogen, for example, the difference between ionic bonds and covalent bonds. We already mentioned hydrogen bonds in the context of water. If you have any deficits in those particular areas of that real basic chemistry, you should be reading chapter 2 in your textbook. But I'm going to kind of assume that you understand that kind of basic concept of how molecules interact with one another through chemical bonding. Not to the extent you have to know it in general chemistry. I'm not worried about all the orbital configurations and things like that. But these basic ideas of a covalent bond, for example, versus an ionic bond. So why is carbon so special? Well, if we take a look at carbon, carbon is kind of an interesting molecule. We've already talked about hydrogen over here. Hydrogen consists of a single proton and a single electron. What about carbon? What's carbon got going for it? Well, take a look at the periodic table up there. Atomic number 6. What's that tell you? 6 protons. And then in its elemental state, how many electrons? 6 electrons. Okay. I lied. I am a little bit interested in total number of electrons that some of the valence shells of atoms can hold. The first valence shell has the ability to hold how many electrons? 2. Okay. For most purposes, particularly in the case of these big 6, what does the next valence shell and all the outer ones hold maximum? 8. Okay. Are molecules stable if their outermost valence shell is incomplete? No, they're unstable, right? So if your outermost valence shell is incomplete, you're going to tend to be reactive. And if you're in the vicinity of another unstable atom, what are you going to do? You're going to tend to try to interact with that atom, i.e. bond, to become stable. Okay? And you can do that by stealing electrons away. What kind of bonds do those form? Ionic bonds, or you can share electrons. That would be covalent bonding. Okay? So this particular orbital of hydrogen is not stable. It has a single electron in that energy level, but it could hold a maximum of 2. Okay? So it could accept one more electron. We say that because of that, it has a valence of 1. Oxygen is a bigger atom. Its first energy level is filled with the maximum number of 2 electrons. And now we go out here to this next level, and it's got 6 electrons. It can hold 2 additional electrons. It could acquire 2 more electrons by bonding. So its valence is 2. Nitrogen has a valence of 3. Carbon has a valence of 4. There's nothing magical about that number 4. But the fact that carbon has a valence of 4 means there's a lot of different possible ways it can interact with other atoms to become stable. There's a lot of different bonding possibilities if you have a valence of 4. One of the things that carbon oftentimes does is to bind with hydrogen. And to bind in such a way that both the hydrogen becomes stable and the carbon becomes stable by sharing electrons. Hydrocarbons are just molecules that are chains of carbon and hydrogen bonded together so everybody's happy and so everybody's outermost electron shell is filled and stable. Hydrocarbons, as it turned out, form kind of the backbone of the 4 major classes of organic molecules. So proteins basically have a hydrocarbon background. And so do carbohydrates. And so do lipids. And so do nucleic acids. But those 4 molecules are different from one another, aren't they? They have different properties. Why are they different? Because tacked onto these hydrocarbon backgrounds, we're going to see later on, will be a variety of functional groups. Like an amino group, or a carboxyl group in an amino acid. Or like a phosphate group in nucleic acids. But all the organic molecules basically have this architecture that consists of a hydrocarbon backbone, and then stuck onto that, unique functional groups that define the type of molecule that that is. Functional groups really kind of determine properties of a molecule. The hydrocarbon backbone just kind of positions those functional groups in the proper way. But really, the nature of the molecule is due to what functional groups it has. Well, here's some different possibilities for types of hydrocarbon chains. There's some small ones. There's some big ones. Here's a real simple one, ethane. 2 carbons, 6 hydrogens. If you start looking at the bonding patterns in these, you'll see that everybody is happy and stable. What does a bar right here, a single bond, represent in terms of electrons? Yeah, you're sharing a pair of electrons. Okay? So carbon has a valence of 4. So to be stable, it needs to attain 4 additional. Okay? So what's it do? It shares one with this hydrogen, one with this hydrogen, one with this hydrogen, and one with this carbon. Everybody's happy. Hydrogen has a valence of 2. So it shares with a carbon. Are you limited in terms of the length of these hydrocarbon chains to just 2? No, you could have 3 carbons stuck together in the case of propane, or 4 in the case of butane. Do these molecules have to be linear? No, they can be branched. Okay? Isobutane still has 4 carbons, but it's not a linear straight chain, it's got a branch on it. Do these have the same properties? No? Different structures. They have the same number of atoms, but they have a different arrangement. What do we call these molecules that have the same number and kinds of atoms but different arrangements? They're isomers of one another. Well, here's an interesting little wrinkle right here. What's going on right here? We've got a double bond between the two carbons, not a single bond. What's a double bond represent? Sharing two pairs of electrons. Okay? Is this carbon stable? Yeah, it's sharing one pair, two pair, three pair, four pair. How does that differ from butane? Butene has one less hydrogen. It doesn't need that hydrogen because it's sharing a second pair of electrons with this carbon. So this is called a double bond. Does the double bond have to be here? No, it could be here. Are these the same molecule? No. They're isomers of one another. Different properties. Well, here's where it gets really interesting. These chains don't necessarily have to be linear. They can actually interact with themselves. And this end can interact with this end, forming a ring structure. Do these carbons have to be joined by single bonds within the ring? No, there can be double bond interactions taking place. But the more double bonds you have, the less hydrogen that you have. See the relationship there? It's kind of interesting. This particular idea of a ring structure was, according to history of science, imagined in a dream. And the person who was looking at these molecules couldn't explain their behavior. He knew what the molecular components are, but the behavior of the molecule didn't make sense based upon the molecular components. Because he was assuming that the molecular components were all in a linear chain. And he had a dream one day. And he dreamt that he saw a snake devouring itself by eating the tail. And said, wait a minute, could molecules have these ring structure? I forget the name of the guy. This is, 1700s or something like that. But this inspiration, this dream, he'd been processing it in the back of his mind. And in the dream, it kind of all came together again. But this idea that there's all this variety of ways you can form these hydrocarbon molecules. Sugars will do this, okay? Sugar molecules in solid form tend to be linear. But if you throw those sugar molecules in water and they dissolve, they tend to form these ring structures. And so glucose inside of a cell tends to have this ring structure formula. Well, these serve as kind of the backbones. And then if you start putting, instead of hydrogens, various functional groups on these carbons, now we start getting some interesting molecules. They're no longer hydrocarbons. If we start putting carboxyl groups and amino groups on them, they become amino acids. Or if you put carboxyl groups on them only, then they might be a lipid molecule. But the basic structural diversity is due to the nature of the hydrocarbon chains. So one of the things that we're going to ask you to do is to become familiar with the basic functional groups that define the four classes of molecules: carbohydrates, lipids, proteins, and nucleic acids. And to be able to recognize some of those functional groups and to have a sense of what properties those particular functional groups have.
So when you start thinking carbohydrates, what should you be thinking about? Well, it's got a hydrocarbon background. But then what functional groups are typically associated with carbohydrates? And what properties do they give those particular molecules? What functional groups are associated with lipids, proteins, nucleic acids? So if you see OH as a functional group, you should be immediately thinking hydroxyl. Okay? What's an example? Well, hydroxyl groups are found in a variety of sugar molecules. They're also found in alcohols. What are some of the functional properties? So in the case of hydroxyl groups, they tend to be pretty polar. And one of the things that's important about polar functional groups is that they tend to attract water molecules. They tend to make things water soluble. So alcohol is very water soluble. Why? Because it has a lot of those hydroxyl functional groups. Sugars also have hydroxyl functional groups, and they tend to become very water soluble as well. Another functional group that's going to be important for us is a carbonyl functional group. What's the structure of the carbonyl functional group? It's got a carbon with a double bonded oxygen. Unfortunately for you, it gets a little bit more complicated than that because there are actually two major types of carbonyl groups. There are ketones and there are aldehydes. They both have C double bonded O's, a carbon double bonded to an oxygen. The difference between a ketone and an aldehyde is where in that hydrocarbon chain is that functional group located. If it's at the end of the molecule, it's called an aldehyde. If it's in the middle, it's called a ketone. So, carbohydrates, for example. One of the ways you can define a carbohydrate is to refer to it as a polyhydroxy aldehyde or ketone. So what does that tell you? Okay, well, it's going to have a hydrocarbon chain. There are these chains of carbon and hydrogen stuck together. But instead of being just all carbon and hydrogen, in place of some hydrogens, there are a whole lot of these hydroxyl groups, these OHs. That kind of conjures up an image in your mind. Okay? And then additional to a hydroxyl group, what does it have? It has either an aldehyde group or a ketone group. So it's got this C double bond O. If it's at the end, it's an aldehyde. If it's in the middle of the chain, it's a ketone. There are two categories of carbohydrates. They can be aldoses or ketoses. Aldoses have the aldehyde group. Ketoses have the ketone group. Glucose is an aldohexose. What does that tell you? Okay, it's a hydrocarbon chain. Any idea how many carbons are in that hydrocarbon chain? 6. That's where the hexose comes from. Hexane. And what's it going to have? It's going to have several of those hydrogens in the hydrocarbon chain substituted by hydroxyl groups. And it's also going to have a functional group somewhere in that chain that's an aldehyde group. So if you understand those terms, do you see you can kind of start building a picture of what the molecule looks like? Well, one of the things that we're going to do in lab next week is do some simple chemical tests for various functional groups. And one of the tests that we're going to be doing is called a Benedict's test for a type of sugar called a reducing sugar. So you'll be reading a little bit about what kind of functional groups that involves. So on Monday, we will continue and go through at least the rest of the functional groups so you can kind of see them and see what kind of molecules are associated with. That'll be a good lead-in into lab. So have a good weekend. If you've got stuff you want to drop off for me, please do.